Hi everyone, my name is Orit Uziel. I'm from the Laboratory for Telomeres and Telomerase Research at the Felsenstein Medical Research Center, Institute of Hematology, David Cancer Center, belongs to Bellinson Medical Center at Tel Aviv University. Today I'll talk to you about our studies regarding premature aging following allogeneic hematopoietic cell transplantation. Allogeneic stem cell transplantation is a procedure carried out in cancer patients, those that suffer from hematological malignancies. In the first step, blood is collected, as you can see here, from the donor blood or bone marrow or from the umbilical cord. Bone marrow is removed under sterile conditions. Then the donor blood is processed through a machine that removes the stem cells. You can see here. The rest of the blood is then returned to the donor. This procedure is called apheresis. The isolated stem cells are then stored in a liquid nitrogen conditions and used later on or used immediately. At the next step, the recipient receives high dose of chemotherapy with or without radiation. This is done in order to eliminate his cancer cells as well as his immune cells to allow the body to accept the transplanted cells without rejecting after transplantation. At the final transplantation step, see here, the cells are transferred to the recipient patient through a catheter placed into his blood vessel. The stem cells travel in his blood and reach the bone marrow where they proliferate and take over the whole blood system, all blood cells of the patient. So that the recipient blood is composed of the donor blood only. Chemotherapy accelerates aging. Studies conducted in recent years suggested that survivors of both hematopoietic cell transplantation and chemotherapy exhibit features consistent with accelerated aging. These include older appearance of patients treated with chemotherapy, high incidence of endocrinological, pulmonary, and cardiovascular morbidity, increased incidence of endocrine, pulmonary, cardiovascular, and neurological morbidity, as well as excess of secondary malignancies. In addition, studies reported about high incidence of frailty in childhood survivor from cancer. Cytotoxic therapy triggers an accelerated of the aging process, which is probably mediated by cellular senescence. What is senescence? Senescence is defined as an irreversible state in which the cells stop divide, getting older, so to say, and eventually die. The senescence of cells is translated into the aging of the whole organism. Usually, senescence is caused by the shortening of telomeres. To understand what telomeres are, let's look on the following slides. And this is the DNA, an illustration of our chromosome, and the telomeres form the end part of the eukaryotic linear chromosome. They contain repeats, as you can see here, of TTA, GGG in humans, and form the segment of between 3 to 12 kilobases in humans. To understand their cellular role and function, let's look on their 3D structure that is maintained and regulated by a protein complex named chaterin. Okay, as you can see here, this is the 3D structure. They form a loop called T-loop for telomeric loop, which is maintained by another loop called displacement or D-loop, in which one strand invades the double strand region here To maintain this whole structure, a protein complex named shelterin maintains this whole 3D structure. Here you can see an example of the proteins of shelterin complex, which maintains the whole 3D structure of telomeres. And this is an electron microscopy image of this T-loop at the end of telomeres. Telomeres are getting shorter in each cell division, which is accompanied to a DNA replication. And the process of telomere shortening is shown here on the next slide. It is called the N replication problem. So this is then the replication problem, in which when the DNA is replicated, there are two strands. One strand is called the leading strand, and the other one is called the lagging strand. Now, the directionality in which the DNA is replicated is the same. It always goes from the 5 to the 3' end of the newly formed strand. So in the leading strand, it goes continuously, as you can see here, but in the lagging strand, it goes on segments like you see here. In each segment, there is an RNA primer attached to the template DNA, which is elongated by DNA polymerase. And in the next step, these RNA primers fall down, and there is a ligation between the short segments. However, nothing replaces the fall down of this primer. So there is a lag here. And this is the reason why the telomeres are getting shorter with each cell division. Actually, they are getting shorter here as well. And this is the replication problem. What is the function of telomeres? Firstly, the main function of telomeres is to protect the ends of chromosomes from being recognized as double-strand breaks that are needed to be repaired. I told you about the shelterin complex, protecting and stabilizing its structure. With no telomeres, the identifying of the ends of chromosomes as double-strand breaks would have been identified as a break in the DNA, which would have been resulted in a repair mechanism that includes fusions of the two ends of the same chromosomes or two ends of different ones. Since this doesn't happen, telomeres protects chromosomes from these fusions and thus provides some stabilization to the whole genome, and this is its main function. A part of it, other functions, include the following. It enables a complete replication of the DNA, contributes to the functional organization of chromosomes in the nucleus, participates in the regulation of gene expression, and since it is getting shorter in each cell division, it serves as a mitotic clock, shortens with each cell division, as I told you before. So, as a mitotic clock, it is considered a mechanism that promotes cellular aging. Telomere length is usually used as a marker for aging. Due to its shortening in each cell division, accompanied to DNA replication, leading to cellular senescence and the aging of the whole organism. As an example, this is a graph generated in our laboratory showing that telomeres shortens as a function of age in the general healthy population, as you can see in this line. This is the length of telomeres as a function of age. But when patients are treated with standard chemotherapy, the telomeres are getting shorter even more. And when they are treated with the high-dose chemotherapy, their shortening is even higher and faster. This is just a summary of what you saw before, if in the general population the telomere shortening is about 50 kilobase a year, then in the patients that are treated with standard chemotherapy, the shortening of telomeres is about 250 a year. But those who treated with intensive chemotherapy, their shortening is about 500 base pairs a year. So it is much higher than the normal population, and it is dose dependent. Now let's go to our patients that underwent stem cell transplantation. Actually, stem cell transplantation presents an ideal system to study the fate of telomeres after transplantation. Why? Because the same cells actually grow in different hosts at the original donor and at the body of the recipient. So if we want to follow their shortening, we can compare it to the status in their original donors. This graph is from a previous study of ours, comparing telomere length in the recipient versus the donors at our cohort. As you can see, usually the recipient, the patient's telomeres are shorter than their donors if you compare them. However, this is not always the case, as you can see later on. So, what other parameters we can use to study senescence? One parameter is the I6 levels in the plasma. The other one is the expression of TNF-alpha in white blood cells. People use also P16E4A expression levels in white blood cells and the expression of the ARF gene in white blood cells. The problem is that although all reflect aging, they're not accurate enough and quantitative enough to study aging. These are an example of aging parameters that were used in previous studies. For example, the level of P16E4A was increased in the blood of about 33 women at the age of 50 or less, treated with chemotherapy for breast cancer. Chemotherapy elevated their molecular age in about 15 years of age of normal aging. And similarly, about 176 breast cancer survivors who had received chemotherapy aged by about 3.5 years of age, according to this marker. Now, recently, another marker was explored, namely DNA methylation. It appears that this marker is much more accurate in predicting the biological age of humans. This is the methylation group, which is attached to this position. And about 70 to 80% of the total CpG islands is methylated in the human genome, and most of the unmethylated CpG is clustered in islands at the 5' end of the genes. Now, methylation of the promoter regions of genes usually results in the suppression of the expression of these genes, but here we do not talk about gene promoters. So this is a methylation DNA. It appears that DNA methylation decreases with age, and as I said before, it is considered as the most accurate method for predicting the biological age. The first researchers that established this method was Horvat 7 years ago. He has conducted a very thorough study in which he has identified 353 CpG sites in the whole genome, taken from 82 Illumina DNA methylation arrays database, covering 51 healthy tissues from 8,000 samples. A very thorough study, and he did it by himself. Accordingly, he established a multi-tissue predictor of age that can accurately predict human age with a deviation of maximum 3 years, as you can see here. Later on, Wagner and his group narrowed down these 353 CpG sites to only 3 sites, with an accuracy of 5 years of age. And they established a company named Sigenia, which sells this service predicting biological age. Now, the genes, the 3 genes that contain these CpG island sites are these: ASPA, aspartosylase is used to metabolize N-acetyl L-aspartate by catalyzing its deacetylation. This is the first one. The other one is shown here, TIGA2B, also known as CD41, encodes for integrin alpha chain, plays a role in coagulation. And the third site is within the gene PDE4C. This is a cyclic AMP, a specific cyclic phosphodiesterase, which degrades the phosphodiester bonds. And this is the predictor that they formed. So actually, these are the 3 genes that I just told you, and these are the methylation sites within these genes. So one has to enter the level of the methylation of each gene, which calculate, and the results are given in units of years. So this is the name of their company. Now, the aim of the study I will describe to you were to evaluate the biological age of stem cells transplantation survivors, firstly by methylation assessment, and secondly by several other parameters, as you will see shortly. Our cohort composed of 26 survivors of allogeneic stem cell transplantation with no active disease, and their respective donors, usually sisters or brothers, were evaluated as a median, mostly 3.6 years after a transplantation procedure. So, after assigning an informed consent, we drew blood samples from the study participants, and the biological age of the donor, white blood cells, residing in the recipients was compared with the age of the white blood cells residing in their respective donor. As I told you before, we have the same blood cells growing either in the original donor or in the patient recipient. A part of this, the cellular age of the epithelial cells of the recipients, the patients, was determined as well by the DNA methylation system. And the methodology of the study was as follows. Cellular age was determined by DNA methylation analysis of blood cells and buccal swabs, epithelial cells from the inner part. Telomere length was assessed by Southern blot, and plasma levels of I6 was measured by ELISA, and the expression of P16 INC4A was assessed by qPCR. This cartoon shows the sample processing let's start from right to left. In the right side, you can see a DNA isolation from epithelial cell swab. This was done by one of the laboratories in our medical center. So this was one arm of the study. The same blood sample was used to isolate a DNA, as you can see here. And the rest of the blood was divided to two. One arm was used for RNA, for DNA analysis for telomere length, and the other one was used for RNA to measure by qPCR the levels of P16. In another arm, we took 4.5 milliliters of blood samples, and from which we isolated the serum, and this was kept for future analysis of IL-6. So we have RNA, DNA from the buccal swab, and serum. So this is the patient characteristics. The median age was about 49 years old, ranging from 28 to 71 years. And 58, about half were males. Most of patients were transplanted for acute leukemia, AML, you can see here, or myelodysplastic syndromes. Received myeloablative conditions to eliminate their blood cells, and all of the patients received peripheral blood stem cell graft from matched sibling donors, either sister or brothers. And the median age of the donors, is not shown here, was about 48.5 years. And all patients were in complete remission, part of this one. I'm not talking about the GVHD and graft versus host disease, in which the graft cells attack the recipient body. So this is the results of the biological age of the transplanted patients and the donors. So as you can see here, the age of the transplanted, either by epithelial methylation or by blood methylation, was higher in all pairs compared to the donor age. I just remind you that these were the same sex. In the next slide, you will see it more clearly. For example, this graph shows the difference between the biological age predicted either by blood age, red, or by the epithelial DNA methylation age in blue. And as you can see, in the majority of the cases, the age as predicted by epithelial methylation or blood DNA methylation was much higher compared to the chronological age of the patients. So this just shows that indeed stem cell transplantation results in higher aging, accelerated aging. The difference was about 8.34 years, and the difference between the biological age, the actual age of the cells, and the chronological age of the patient. If we look on the blood cells, it was 8.3 years, and if we look about the predicted age by the epithelium, it was even higher. 8.7. And this summarizes the difference between blood predicted age of the recipient and the donors. This was also high, about 6.3 years, plus minus 6 years. So again, accelerated aging of the recipient. Another way of presenting the results is the average actual age of the patients versus the predicted biological age. So if the chronological age of the patients was about 48.9, then their age, their biological age, was about 58.5 on average. So again, accelerated aging. Another comparison is shown here. This time it is the median age of cells in patients. 58 years of age versus the median age of cells in the donor, which is only 53 years. So again, these are the same cells growing either in the original donor or in the transplanted patient, and you can see the difference in years. So these were the results of the DNA analysis, and here we present the other parameters. You can see here that the IL-6 levels at the serum of the transplanted patients was higher, much higher in all pairs if we compare them to the donors. Again, a sign for accelerated aging. The level of P16 in INC4A relative to the levels of the donors in the patients was always higher, as you can see in this graph. And finally, telomere length. So these are the length of telomeres that show mixed pictures. Some of them were elongated, so the transplanted, for example, here, but some of them were even shorter, for example, here, versus their donors. So we can't say a strict conclusion, but we know that after transplantation there is sometimes a compensation process of the shortening of telomeres. Maybe this was one of the reasons for that result. Another essay that we conducted was an essay for Henry for a 6-minute. These two parameters also present accelerated aging in the transplanted patients compared to the control group. You see that the hand group was to a lesser extent, and also 6-minute walk essay shows that in the control age matched group, their ability to walk during 6 minutes was much lower in the transplanted patients, again pointing to accelerated aging. So to conclude our study, we can say that these results suggest that allogeneic stem cell transplantation is associated with significant accelerated aging of both the hematopoietic and the somatic systems. Somatic systems is the epithelial tissue. And to the best of our knowledge, this is the first study employing these novel markers for the assessment of accelerated aging in a clinical setting. And I would like to thank all the participants in the study. First of all, Piera Nani, the head of the Institute of Hematology in the David of Cancer Center, Anat Be'eri from our lab, and the German group, Sigenia, for measuring the DNA methylation levels